Okay, in this class we discuss about databases. So, do you remember what did we discuss in the last class? About the DNA and RNA. What is the difference between DNA and RNA? Deoxyribose and H and OH groups, right, at the 2 prime side, and then the base side. So, you can see the thymine and uracil, right. Right. Then we discussed about the complementary strand. If you have a DNA sequence or RNA sequence, you can get the reverse complementarity, right. That will do that. And then we discussed about transcription and translation. So, to convert the DNA sequence to protein sequence, and also discussed about the software. Which module or which suite we discussed? Emboss, right. So, Emboss software we discussed, and there are a lot of options available in Emboss, right. So, we use RevSeq, right, for this complementarity as well as the protein sequence. Then we discussed about reading frames. So, how many reading frames? 6, right, 3 forward and 3 reverse reading frames, and we discussed about few of the sequence based parameters, specifically on the stiffness or the flexibility or the base stacking energy, right. So, if you discuss about the major aspects of bioinformatics. So, the first class we discussed about 5 major aspects of bioinformatics, right. What are the 5 major aspects of bioinformatics? Databases, and then get the hypothesis, and development of tools or online servers, and virtual screening of compounds, and currently bioinformatics is used in the next generation sequence analysis, right. So, the first one is the databases. So, in this class we will mainly discuss about the databases. So, what is a database? Yeah, structural collection of information in organized form, right. So, if you look into this literature, there are many databases available, right. For example, if you use the general search, right, which one you use? Google, right. You go whatever information you want, immediately go to Google and type, and then you will get the information, right. So, likewise in the case of the bioinformatics, right, what is the major field in bioinformatics? Biology, right. Biology will produce a lot of experimental data. So, we look into the biology, right, we can get the experimental data on various aspects, specifically on the macromolecular sequences and the structures, expression profiles, pathways, and so on. So, we have a wealth of data available in the literature. The experimentalists, they create the data and they publish in the journals, right. So, these data are available in the literature, right. So, it is very important, right, and necessary to collect all the information and put it together, right, in a computer readable form. Right. Earlier days we used to collect the data and keep by ourselves. Currently, due to the advancements in the computing and the transferring files and all, it is possible to share the data, right, to public. So, likewise if you see the data which are scattered here and there, that means the published in different journals articles. So, it is a pain to collect all the data, otherwise those who want to do the analysis, they have to collect the data. So, it is time consuming and everyone has to do that, right. So, if you have a database and the collection of data in one place, then it is easy to extract the data and use it for the analysis or the development of any servers. So, here it is scattered in the literature. So, you put it in a ordered form, right, and a few years ago they put together in a form of book. So, they collect the data, for example, the thermodynamic data, Fail collected more than 22,000 data and publish a book. So, if you have a book, then again we need to look into all the details and try to make into the computer readable form, then there are a lot of errors, the possibility of errors. So, it is important to have the organized collection of this information and a computer readable form, because in this case we can reduce the errors, right, and easily you can use the information for the further applications, right. So, for example, if you want to develop a database, right, what are the major characteristics you like to do?
First we have to reliable content.
Yeah, first you have to fix about the contents, right. So, who will use the contents, right, how shall we get the contents, right, who are the end users, right. First you need to decide about the contents and the importance as well as applications, right. If you develop a database and nobody is using, right, then there is no use to develop such database and putting so much of pains, right. So, if you develop a database and that is useful to many others, right, then it is very important. So, that will be very helpful for many researchers, right, fine. So, one is the contents, then what is the other aspects you wish to consider?
How to search in
Yeah, we need to think about the users perspectives, right, whether the users like to get the data on which aspects, right. For example, if you type something in Google, if you want to get any information from Google, if you type, so will you get the first attempt or you take many attempts? Mostly the first attempt. Sometimes it will take time, it will you have to search again and again, right. But what if you search 10 times and even 10 times you do not get the data, what will you do? Will you use it again? We will not use it, right. So, the thing is, when we search the user's perspectives, the user should get the reliable data and the required data, that is important. And the second thing is the time, right. Now, the world is moving very fast, right. So, even they do not wait for even 10 seconds, right. If you Google in any of the information, if you Google takes 5 minutes, right. So, will you use that to Google again? We do not use it, right. We expect the result in 1 second, as soon as we put enter, right, then you should get the result, this is what they need, right. So, the time, that is very important. And what else we need to think about? Yeah, how to get the display the data, because as per your requirements. Then the second thing is you need to get the reliable information, right. Now, if you get lot of WhatsApp messages, right. So, more than 70% are not reliable, right. Whatever people think they make and then they send as viral. But if you get the required information and reliable information, then you can trust. If you do not get the reliable information, if you Google it and if you get lot of random information, then it is not useful, right, because you cannot trust. So, whatever the database you develop, the data should be reliable, right, that should be user friendly and there should be reliable information. Then what else you need to think about? It should be less redundant, because if you have the same data, if you repeat again and again, then that will be boring to the users, because they have to do lot of work, right. So, that should be less redundant. So, you have to give the high integration and less redundant, right. Then what are other aspects you used to think about?
Understandable.
Yeah, it should be understandable, right. We have to give some information, you are familiar with all this information, but the users are not. So, in this case you have to make in such a way that the user should understand your data, right, that is very important. Then what else? Yeah, some sort of applications and references, as well as you should give an option to download the data, right. Otherwise, if you guys display lot of information, it will be difficult. For the case of the literature it is fine, but the case of the biological data, so it should be downloadable, because we have deal plenty of data. So, editing is again it is an issue. So, we can give it as to download the data, then several users they use that database, fine. So, likewise, if you look to develop a database, you have to think about various aspects. On these aspects, if you consider and develop a database, then you will get lot of users, right. Many people use it and then there will be a famous one, right. Okay. So, now I will discuss some of the important aspects and what are different characteristics of a database. Some of them we already discussed, right. Some of them I will go through some points. First one is the contents that we discussed earlier. So, contents is very important, right. There are several databases available in the literature. So, could you list some of the biological databases? PDB, protein data bank, right. If anybody wants to have the structures, right, immediately they have to look on the PDB, because that is a unique source, you will get reliable information. Then what else? NCBI, like the UniProt for the sequence database, thermodynamics we go to Protham, right. Our lab also we developed several databases for example, Proximate, the binding affinity of protein complexes, right. So, in all the databases the contents are very reliable. So, the contents are mainly the experimental data, right, and give you the sufficient information to the users, right. This is what the users require, right. So, contents is very important. And the second one is the ontology, because in all databases, because they have to develop with several constraints. So, they use various keywords, right, they use various terms and conditions, right. So, whatever the biological term they use, right. So, in this case they have to give the details, right. For example, you put secondary structure, right, you know secondary structure, but we cannot assume that everybody will know the secondary structure. So, you have to explain at least in one line, you have to write what is secondary structure, what are different secondary structures you consider in your database, right. Solvent accessibility, right. The biologists, some of them are familiar with the term, some of them are not familiar. In this case, you have to explain, what is that and what are the various terms we consider to define the solvent accessibility. Fine. So, for all the terms we used, you have to give the definition, right, and the third one I put the logical structure, right. So, what is the main aim of your database? For example, if you take the proximate, it is a binding affinity of protein protein complexes, that is the major goal. If you talk about the PDB, the main aspect is the 3D structures, the coordinate XYZ coordinates. If you take the protham, so the major aspect is thermodynamic data for the proteins from folding to unfolding as well as for the mutations, that is a major one. Now, we have to supplement the data with the other information, right. If you talk about the thermodynamic stability changes with the experimental conditions. So, you have to provide the conditions, what is the temperature, what is the pH, and about the buffers, ions and the concentration and so on. And again we can give more information regarding the structures of the protein or more detail about the proteins as well as the mutations and various structure based features, right. All these things we need to give, right. In this case you have to frame a logical structure, right. Okay, the major thing is your data and how to supplement with other information, right. So, the fourth one is the format of the data, because for each entry you give some information, right. If you use various format, then the users will be confused. So, you have to follow a uniform format, right. If you look into the UniProt, right. So, if you see the first line is the name and the synonyms and all the order is the same. If you go to the protein data bank, so you give the compound and the protein name and the resolution. So, you see the particular order, right. So, in this case you have to give your data in specific format, right, that is very important. I will show some of the details, right, in the later slides. Then now you have the overall view of your data, right, what you want to put in and what the terms you use and how to design your database and how to format your data, right. Now you have the data, then put it in the website, then it should give some sort of sources to retrieve the data. So, you can give some search options, right, this will be give flexibility to the users, right, because all the users they do not want the same data, right. Some of the users they require some data, some they require they think about the different types of data, right. So, in this case you need to give a route to select the data from your database and then you have to present the results, right. If you present everything together there will be a chaos, right. You have to present in a way that is very legible and understandable to the users, right, also a requirement of the users, right, this is fine. So, then the data is there now. Now, the issue is your database it contains various other information, right. In this case you have to provide the information where you link the data. For example, your main aim is thermodynamics, but if you link with the structures or the sequences, then you have to give the link of the structural data or the sequence data, right, you have to give all the information, right. Okay, now we will explain with some examples. This is a database called thermodynamic database for the proteins and mutants that we developed a few years ago, right. So, first we give the contents. So, this is the one we give the details about the contents. So, we give sequence structure information, right. If you go to the website you will get all the details and the thermodynamic data and the experimental conditions, literature and so on, right. And the second one I show the first one is the contents and the second what is second aspect? Terms and conditions, ontology, right, the terms and conditions. So, if you see these are the various terms we use, right. So, for all the terms we use we need to give the details, right. For example, it is a PMD number. So, PMD number means okay we know what is PMD, but the users are not familiar with the PMD. So, this is protein mutant database number, there is a database called protein mutant database, right. So, here we give the expansion. The for example, PDB wild. So, what is PDB wild? PDB wild means protein data bank for the native protein, because this database is for the mutants. So, here we give the mutation information. So, for this case you have to give the information, what is the first one, what the second one, what is the number, right. Here in this case lysine K is replaced to proline at residue number 60, right. So, we give all the information in the ontology, right, then the users know. So, what is mutated to what, otherwise this will be difficult to understand, right. And what is number 3? We have the schema, right. So, here I give the schema. So, this is our central thing, our theme is thermodynamic data. So, Pratham is here, right, this is the central theme, and here we give the data on the experimental conditions. So, I give thermodynamic methods and conditions and the literature information, right, and the data. What are the data you put in the program? So, we put the delta G, right, free energy change, delta H, delta Tm and the delta G H2 and so on, right. This is the free energy change due to the thermal denaturation or the denaturation, melting temperature and so on, right. Fine. Now, we give the sequence information, structure information. So, I put the data here, we give the sequence information, right, PIR and Swiss prot, currently they merged to uniprot, right. Right. So, here structure is PDB and the function say the motif, mutation and so on. Now, we give the other databases, right, for to link with this one, like as the Brenda database, this is for the enzymes and the PubMed database, right. So, now we give the interface so that connect to the internet. So, you will get the data and you can download the data and you will use it for the further applications, right. This is what we do, right. This is one is the contents, second the ontology, there is a schema, and the fourth one specific format, right. So, you give the format. So, we use a specific format for the database. For example, start with the number, that is our Protham number and the protein name and the source and the mutation. So, they follow the same format for all the data. So, currently we have more than 25,000 data. For all the 25,000 data, we have the unified format. First line means the line contains a number, 10th line means I have the delta G. So, we have the same information, same line. So, easy to search and interpret the data, right. Then we provide options, because users have various options to obtain the data, right. So, here we give the search options. So, users can obtain the data with various search conditions, right. In the future slides I will explain the details. Right, here we give a lot of conditions. So, users can use any of these conditions, right, with and or right, to obtain the data. So, when they get the data, right, they click the start button and they will get the results. Here I searched with the lysozyme, right, this, so we get the data with the lysozyme, right. So, we have various other conditions. What are the data which fulfill these conditions, we display the results. Then we give the link. For example, here I link with the reference, this is the article, right. So, if you click here, so they will get the literature. So, where you get the data and how they use the experimental conditions, all the information you can get from this PubMed database, right. So, there is a complete database, right, which contains the information regarding the thermodynamic data supplemented with other information with proper search option, display option and download option, right. And then other aspects we discussed earlier, it should be very fast, should be available all the times, right. If you search any of the websites and it is not available for 3 times, then nobody will use, right. At least if your server is down, then you should give a caution that currently server is down, it will be available soon, right. Otherwise people think they are not maintaining the database, right, that is very important. Fine. So, now how to develop a database, right. So, there are various ways to develop a database, right. So, in 1970, so F. Card from IBM, he described about relation database, right. So, what is a relation database? What is a relation database? It is a database which gives a correspondence between different features in the database, right. So, this basic unit is called tables, right. So, what they do, they put different information in a table. So, these different records are called attributes and these attributes are interlinked with each other. So, without rearranging this table, this information, you can fetch the data, you can extract the data on any aspect, right. Also, there are several operations you can use. You can use the intersection, union and the difference and so on to facilitate the processing of the very complex queries. I will tell you one of the examples. Okay, so, we see the table 1, right. What are information available in table 1? Amino acid, the name, 3-letter code, 1-letter code, volume, surface area and distal group, right, which groups, right. So, methyl group or guanidine group and so on. Now, the question is, what are the 3-letter codes of amino acids which have distal carboxyl group? So, what to do? Go to distal group, right. The question is carboxyl group. So, what are carboxyl group is here? So, here is the carboxyl here is the carboxyl, right. Now the question is what are 3 letter codes of amino acids, then where you have to look, 3 letter code, second column, right. So this is aspartic acid and glutamic acid, right, that is fine. Now the second question, if you see, it is a compound question, the same question, what are 3 letter codes of amino acids, right, with volume more than 125 angstrom cube, right, and they have a distal carboxyl group. So what is the answer? The volume should be 125. So which is the answer? This one, right? This is more than 125. Fine. So this is you get the information from table 1. Now next question, what are 3 letter codes of amino acids which can serve as hydrogen donors? This information available in table 1? No. This information available in table 2? Donors, yes. 3 letter codes? No, right. So in this case you have to use tables 1 and table 2, both the tables you have to use, right. First go to table 2, this may be considered table 2. So guys, hydrogen donors. So what are hydrogen donors? For example, take this, yes, right. So and the amide, then go here with the amide, this amide is here and the 3 letter codes is this one, right. So here you need to join. So if you natural join you can do table 1 and table 2, if n elements in table 1 and n elements in table 2, so you get the product is n elements, right. So here 20 amino acids and 12 distal groups, so totally around 240 rows, right. So for example alanine, so this is the 3 letter code, this is the 1 letter code, this is the volume, right, you can see the group, and this is from table 1 and this is from table 2, right. You merge these two and you get the information, right, fine. Now what will happen if you go through the complex queries? For example, the question is you have to get 3 letter code, okay, that is fine, with volume between 100 and 150. So second question, right, and the third again that can serve as hydrogen donors and not hydrogen acceptors, or they have surface areas greater than 120, right, and the distal methyl groups. It is very complicated. So we cannot easily see the tables and then get the data, right. So here we use the structured query language, that is called SQL, right. It is well standardized, right, the language. We can get the any complex queries, you can get the data from a relation database, right. So here this is the syntax. First it is 3 letter code, that is the code you need, and the conditions from where, the side chain volume between 100 and 150, okay, this will fulfill these conditions, right. Hydrogen donor, yes. Hydrogen acceptor, no. And then what is next condition? Surface area more than 120, right, distal group methyl, right. Now it is easy, right, you can use the syntax to fetch the data from this table, right. So you can go to any complex queries, right, you can write a syntax and you can get from any relation database, right, fine. So now we show some places you can get the data together. So there are several databases available in the literature, right. For if you look into the nucleic acid database, right, issue. So the nucleic acid research, they publish one special issue every year. The first issue of the nucleic acid research, right, in January, first issue, they publish the databases, right. So if you look to this NAR website, and then there is the listing of databases, right. Currently there are a lot of more than 500 databases listed in the NAR database list, right. And if you go through this database, you will get lot of information, right. Also they classify the data into various aspects, right. These are the different categories, right. So they classify the data based on the nucleic acid sequence databases and RNA sequence databases, protein sequence databases and structure databases, thermodynamics and genomics and metabolic and signal pathways and human gene and diseases, proteome databases as well as other molecular biology databases, plant database, immunol database and so on. So they have different categories, and if you are interested in protein structures, you go to the structures, and even if you go to protein structures, right, there are various sub classification, Right. What is the major database for protein structure? PDB is a major database, right. If you did PDB is a major database. Based on the data available in PDB, there are various sub classifications. So, can you tell some classification from PDB? PDB-TM. What is PDB-TM? Transformed proteins, right, the database of transformed proteins. Also you have CATH, right, and SCOPE. This is for the structural classification of proteins. And Astral, what is for Astral? This is non-redundant set, right, you can get non-redundant set of structures, right, using Astral. So, you have several databases. So, for each category, right, for each category is listed in this database listing, there are several sub classifications, right. You can search this listing and you can find the data what you want, right. If you go to the thermodynamics, again you have several classifications, plenty of sub classifications, and you go into the details and then see the database available. This will help in different ways. For example, if you are looking for any specific data, you can go through the database and check whether the data are available or not, and you come to the data. Second aspect is, if you want to develop a database, right, first you have to check this should be unique, this should be novel, right. Already if your PDB is available, you cannot make another structured database, right. In this case you need to think and check whether such database is available already in the literature or not. If it is available in the literature, how far your database will be unique from the existing ones. If not, then what are the applications, who are the end users, right. So, you have to think about all the details, right. Even if it is available, then you can think who are the users, how many citations they got, right, whether still they are continuing or they stopped in between. So, all the information you have to collect, and based on the available information you have to develop a new one, right. In this case, this is very important, you have to look into the details of the data available in the literature, right. Then this also help you to design your research problem, right. For any bioinformatics or computational biology problems, you need to have a sufficient number of data, right. If the data are not available sufficiently, then your databases are not sufficient, right. If the data are not sufficient, then we develop a model may not be significant, right. Whatever the hypothesis or the models you make, they are not significant. So, you have to get reliable number of data. So, you can check the literature and mainly the databases and see whether you can get reliable number of data, right. For example, if you are working on the binding affinity of protein DNA complexes or protein carbohydrate complexes, right. For protein we have sufficient number of data, we can do it. Protein nucleic acid compared to protein less number of data, carbohydrate even it is very less, right. So, check is available or not, if yes, find the data where you can find, if not, make a new database and first see whether you can get sufficient number of data. We once you make a database, as I discussed earlier, so we should have reliable information, sufficient number of data, should be many users. So, various other aspects you have to think before you develop a database.